Hi everybody, Dr. Tucker, Lab Coordinator, back again. So I know you guys have been using simulators and virtual labs and whatnot the last few weeks. I looked all over the place and I couldn't really find a good simulator for this week that really kind of approximates what we do in class. So I thought I'd head back into the prep room here, go ahead and do the experiment and walk you guys through it. So this week we're doing cellular respiration, as you know. We're going to do two parts. One, we're going to look at aerobic respiration using germinating mung beans to see how that works in the presence of oxygen. And then another part, we're going to look at anaerobic respiration using yeast, who does it in the absence of oxygen. We're actually going to start with the yeast first because they have to run for longer. So I'm just going to walk you guys through. Take a look in your lab manual, obviously, and you'll be able to see exactly what the process is. So we are going to skip part B. We're just going to do part A, the different substrates. We're going to skip the part about inhibitors and cofactors, and activators, they're also called. So looking at part A, we are just going to aim you down here. So the different substrates just refers to different sugars. So yeast eats different sugars, carbohydrates. So you should remember some of this stuff from carbohydrates if you can remember carbohydrates, which seems like forever ago. So our list of substrates that we have are glucose, fructose, maltose, sucrose, starch, and water. And of course, water is acting as our control. We also have a yeast solution. So just plain old yeast like you would get in the packets at the grocery store. And that's 3 grams of yeast that's been dissolved in 100 milliliters of water. Now yeast is also known as sucrase. If you remember from our enzymes lab, the sucrase is the enzyme that breaks down sucrose. So yeast and sucrase is synonymous, so you might see that in the lab manual, going back and forth between yeast and sucrase. All right, so I am just going to set this up, and we're using 2 kinds of respirometers today. This one, I have gone through and labeled each of these small test tubes 1 through 6. Maybe I can put something white, you can see the numbers a little better. And I labeled the big test tubes with those same numbers. Now in your lab manual, it will show 1A through 6A. Since we're not doing part B, I just labeled them 1 through 6. So what we're going to do is we're going to take these small test tubes, and we're going to put 3 milliliters of each substrate. We're going to start with glucose. That goes in number 1. I've got number 1, the small test tube. I'll put my 3 milliliters of glucose in there. Rinse my graduated cylinder. And I'm going to put 3 milliliters, this is just water. And then for each of these, I'm not going to do it just yet, we'll end up filling the rest of the tube with the yeast solution, the sucrase. So I'm going to go through and add all of the different substrates to each of our 6 small tubes, and I'll be back and show you what that looks like. Okay, so I've filled all of these with the different substrates. You can see they're all full to about here. Now I'm just going to top each of these off with our yeast solution. Stir. I'm just going to use a pipette to fill it up to the brim. Show you guys as best I can here. A little bit more. As high as I can get it, all the way up here. And then I'm going to go ahead. I'm going to put the bigger test tube with the same number on top here. You can use a pencil or something. Push it all the way up in there. and then really quickly flip it around. So you see it only loses a very slight amount that you can see down in the bottom there. And the rest, you can see there's a little bit of an air bubble there. Now what we're going to do is we're going to do that with all of these different ones. And then we're going to stick them in a warm water bath. It's just 37 degrees, so it's body temperature, or typical good temperature for yeast. And we're going to wait and we're going to see, we're going to test every so often to see how much respiration has gone on. Now, before I give you the data here, you guys should go ahead and make a hypothesis about all of these different substrates that we have. Again, I'll show you those. Which of these do you think is going to respire the most? So when the yeast eats those carbohydrates, that's when respiration happens. So think about which one of these you think might show the most respiration and make a hypothesis about that. So I'm going to go ahead and do the same setup on all of these different ones and get them in the warm water bath. Okay, so I have all of the tubes filled and inverted. Now what I'm going to do is just go ahead and measure this bubble. You can see there's a little bit of space in the small tube. And I'm going to go ahead and measure that using a little millimeter ruler. And I can tell how much respiration is happening by the size of this bubble. So I'm going to tell right now this little bit of space is just from what came out in the bottom. But as respiration happens, CO2 is produced, and it's going to push more and more fluid out and create a bigger and bigger bubble. So I'm going to measure this, put it in the warm water bath. I'm going to measure it every 15 minutes, and I'll record that. And then we'll be able to tell which of these different substrates has the best respiration for yeast. Now, because we're scientists, and normally if you were in class, you would put all of your class data together because replication is important in science. I've gone ahead and I have actually 3 replicates of this experiment. So I'm going to put all of them in this warm water bath, and I'll get that started once I measure them. Okay, so I've got all of our tubes in the warm water bath. I measured that bubble, so we'll be able to determine exactly how much respiration is happening. I'm going to measure those every 15 minutes for an hour and a half. And I'll give you those data, but you should use those data to fill in the table on page 112 of your lab manual. And you see I've got mine started for all 3. So now we're going to move on, really kind of go backwards to the first exercise where we talk about aerobic respiration, so in the presence of oxygen. Now to do that, we're going to look at beans or seeds. These are mung beans, a type of seed. Now seeds are just the embryo of a plant, so it's plant babies, but they're dormant and they wait until it's a good time for them to germinate and create adult plants. So what we're going to do is we're going to test how much germination is happening in mung beans. Now some of them, we're going to have 2 treatments. So I just showed you these. These are your hard kind of beans that you would find, dry beans. Some of them have been soaking overnight, and you can see they're a lot more plump. So you might know this from life, that when you want to germinate seeds, it helps if you give them a lot of water because they need to take in water to break the seed coat. Everything that a seed needs to start growing into a plant is contained within that seed. It depends on what kind of plant you have. They'll either use endosperm or cotyledon as food before they can start photosynthesizing because they need their leaves and chloroplasts and all that. So we are going to go ahead and use a different kind of respirometer to determine which of these seeds is going to respire more. Now before we start, again, go ahead and make your hypothesis. You think that the seeds that are dried are going to respire more, or the ones that have been soaked? So what we're going to do is use this different respirometer. So this is a respirometer here. It's just a test tube with a stopper and a pipette. Now this is open on the end. So what we're going to do is we're going to put in a little piece of cotton, and to this cotton we're going to go ahead and put a few drops of potassium hydroxide. Now potassium hydroxide just takes any carbon dioxide and converts it to a solid, so it won't take up space. So what we're going to do is we're going to put these in water. This is a water bath. I don't know if you can see it. And because it's a closed system, a little bit of water will come up into this tube, but it'll stop. Now, once respiration starts happening, we know that the beans will go ahead and they'll start producing some CO2. Now, that potassium hydroxide is going to turn that into a solid, so it's actually going to start reducing the amount of air that's inside this test tube. So water will start creeping in, and that can tell us exactly how much respiration those seeds are doing. So I'm going to set this up a little bit. I'm just going to put several drops. This is actually kind of dangerous stuff, so if we were in lab, your lab instructor would tell you to be very careful. I'm going to put a few drops right onto this cotton. Now, like I said, it's kind of dangerous stuff, so it wouldn't be good for our seeds to come in contact with it. So I was very careful not to run it down the sides, but I'm also going to put a different kind of cotton or rayon that's non-absorbent. So it's going to act as a little bit of a barrier between the actual seeds and that potassium hydroxide. Just gently stuff it in there. And then I'm going to take $20 seeds of each. And just put them right in. Try and get most of them in there. Go ahead and put our stopper on gently, and that's it. Then each of these that I do, so this one's going to be the ones that were not soaked, I'm going to go ahead and I'm just going to put them in the respirometer like this and into our water bath. I'll leave it kind of angled upward like that so that it can get used to the water temperature. So it's going to equilibrate to that water temperature. I'm going to give it 5 minutes to do that. And then I'm going to go ahead and put them down in the water. So I'm going to set up the rest of these, and then I'll come back and show you when I put them down in. Okay, so I have filled all of these. You can see, again, we're scientists. We need replicates, so I have 3 of each treatment. So we've got the non-germinated mung beans here, and then we've got the germinated mung beans here. You can see there's a big difference in size of those beans just from soaking overnight. So I'm going to go ahead and I'm just going to rest them on the tape here. Like I said, I'm going to let them all equilibrate to the temperature of the water for 5 minutes. You might be able to see, maybe pick it up and show you, that I've added some food coloring to the water, and that just makes it easier to read the readings later on. So I've got them all sitting in there. I'm going to give them 5 minutes, and then I'm going to go ahead and just move them down off the tape and set them right in. We'll get a little bit of that bubble moving in, and then I'll make my initial measurement based on that, and I'll come back and show you that. Okay, it's been 5 minutes, so everything's equilibrated. I'm going to go ahead and just pop these down into the water. I'm going to make sure that I can actually read the pipettes from where I have them. I don't want to touch them once they start going here. This one's longer. try and keep them, they're a little bit floaty. Try and keep them so I can read them. Now you can see a few of them have already started, and you can possibly, I don't know if you can see that, but a few of them have already had a little bit of the water start coming into the pipette. Now we're going to wait 5 minutes, and we're going to check that and measure it every 5 minutes for an hour. Okay, so our 90 minutes is up on our yeast, and I have been measuring every 15 minutes, and I'll send those data out to you guys, and you can use that to fill in your tables and all that good stuff. So I just wanted to show you, see if I can show you here. You can see, I'm focusing very well. Let me put something behind it. I don't know if that helps. Yeah, maybe not. But you can kind of see here where it's all the way down here. So it's respired enough to create enough CO2 where it's pushing the liquid down. Now the one next to it, this one's the water, so it's our control, and it's barely moved at all. It moved a little bit, but not a whole lot. So you can see like the different substrates will have different effects, and the yeast will eat more of them essentially and produce more CO2. So like I said, I'm going to go ahead and I'm going to put all those data together and send them out to you. I can try and show you our other respirometers here. These are moving along and they're almost done. I've got one more reading to take and they'll be all done, and you can't see the bubbles, but they have moved down as the seeds have started to respire. So same thing, I'll send you guys all of those data, and there's replicates of each so you can take averages so that you can try and get at some trends, because every now and then, you know, a treatment might not be correct. So that's why we do replicates and we take averages. Okay, so you can use these data, fill in your tables, answer the questions in your lab manual, and just submit those. Again, if you don't have a lab manual, I've been posting the chapters on the weekly assignment, so you can find all of it there. Okay, thanks guys.